This is the third video in the unit on genetic variation. In this video, we will discuss the connection between DNA mutations and the genetic variation observed between individuals within a population. In Chapter 14, we discuss the formation of mutations in a DNA sequence. It is these mutations which cause slight differences in the DNA between individuals in a population. Those differences, such as the C and T highlighted here, are called DNA polymorphisms, and they produce genetic variation within a population. When we examine an individual's genetic information, which is the order of nucleotides in their DNA, we are examining their genotype, or what the gene says. As we discussed in Chapter 14, some mutations in DNA can affect how the encoded protein functions. When protein functions differently, this will lead to changes in an organism's phenotype. The phenotype consists of an organism's observable characteristics, such as eye color or height or fur color seen here. Keep in mind that when you look at the genotype of an individual, the individual has two copies of each gene, one from the mother and one from their father. Because of polymorphisms, those different DNA spellings caused by mutations, there are oftentimes more than one version of a gene in a population. For example, if we look at the same gene in a different individual, they might have a different spelling. We can see here that individual 2 has a cytosine in the DNA sequence, whereas individual 1 was carrying a guanine. The different forms of any gene are called alleles. Notice that while individual 1 and individual 2 have different alleles compared to each other, individually their two copies of alleles are the same. These individuals would be described as homozygous. An individual is homozygous for a trait if they have inherited the same type of allele from both parents. If an individual inherits different alleles from their parents, as seen here in individual 3, they are now considered heterozygous for that trait. In Chapter 16, we will learn how to identify what the phenotype of an individual is based on if they are homozygous or heterozygous for a particular gene. Often people think of a mutation as something that must be bad. However, this is not the case. You can have harmful mutations, which have a negative effect on the organism's survival, neutral mutations, which do not affect an organism, and beneficial mutations, which can increase an organism's ability to survive. This classification is dependent on both the genotype and the environment, because both genotypes and the environment influence an organism's phenotype. For example, the HbA allele encodes the normal wild-type gene of hemoglobin, which carries oxygen in the blood, while the HbS allele contains a mutation. But is this mutation beneficial, neutral, or harmful? If someone is homozygous for the HbS allele, they will develop sickle cell anemia because they only produce HbS hemoglobin, which cannot transport oxygen well. This can be fatal, thus it is a harmful mutation in that instance. However, if someone is heterozygous, they will produce some normal functioning hemoglobin and some sickle cell hemoglobin. In this instance, the amount of normal hemoglobin is often enough for the organism to function properly, making the sickle cell mutation neutral if only one copy is present. Interestingly, if this individual lives in an area where malaria is carried by mosquitoes, the presence of the sickle cell mutation becomes beneficial because the sickled red blood cells help prevent the person from getting the disease. Oftentimes it is beneficial to be able to tell the difference between the DNA found in two individuals, such as in testing of DNA at a crime scene. DNA sequencing, to look at the exact sequence of nucleotides, is too expensive, so instead people rely on a combination of other methods. Two in particular are called the variable number of tandem repeats, or VNTR, and restriction fragment length polymorphisms, or RFLP. VNTRs look at differences in DNA based on insertions and deletions that occur in certain hotspots of the genome, while RFLPs allow you to examine base substitutions that may differ between individuals. It is the VNTR method that is used in DNA forensics and paternity testing.